Hello, and welcome to Development and Validation of a Platform Assay Method using Variable Path-Length Technology for Oligonucleotide Drug Products, presented by American Pharmaceutical Review and sponsored by Repligen. My name is Mike Auerbach. I'm the Editor-in-Chief of American Pharmaceutical Review, and I'll be the moderator during today's event. In this webinar, you'll learn the benefits of implementing the CTech SoloVPE using variable path-length technology as a platform assay method for GMP manufacturing of oligonucleotide drug products, how to overcome challenges due to unique characteristics of oligonucleotide and optimize the method to achieve desirable accuracy and precision, and how to validate the method in accordance with ICH guidance and utilize measurement system analysis to further access the method capability. In addition, this event will hold a live question and answer session at the end of the presentation. To ask a question, click on the Ask a Question tab on the right side of your screen. Please take note, the right side of your screen also features an overview of this webinar and more information regarding our speaker. If you have a technical question during the event, click the Test Your Connection button at the bottom of your screen. From there, you can access additional webinar support. Finally, we encourage you to use the social media widgets beneath the webinar to share with your friends and colleagues. And now, allow me to introduce today's speaker. Hai-Ching Yin is the Assistant Director of Pharmaceutical Development for Ionis Pharmaceuticals, responsible for development, transfer, and validation of the platform SoloVPE method to support multiple early to late-staged antisense oligonucleotide drug products manufactured at CMOs. He has been with the company for more than 2 years, with over 10 years of industry experience in oligonucleotide and small molecule drug product development, specialized in applications of DOE and other statistical tools in formulation and process development. He received his PhD in physical chemistry from Peking University of China, followed by a postdoctoral training in pharmaceutical science from the University of Utah. Hai-Ching, welcome to the webinar.
Thank you, Mike, for the introduction. Welcome everyone for today's webinar. So in today's presentation, I will start off with a brief introduction to antisense oligonucleotide therapeutics, talk about why we need in-process assay control and the challenges for method development, and then I will elaborate on how we were able to develop a suitable method and get it validated and transferred to CMOs. I will also talk about how to utilize measurement system analysis to further evaluate method variation and touch base on some future applications. And finally, I will provide a brief summary. Antisense oligonucleotide, or ASO, belong to an emerging new class of drugs known as oligonucleotide therapeutics, with capability to treat a wide range of diseases, including a lot of rare diseases with great unmet patient needs. An ASO molecule is a short single-stranded hybrid DNA and RNA sequence with a molecular weight of around 7,000 Dalton. It is chemically synthesized to target a specific mRNA. As shown in the cartoon on the right, once ASO binds to the complementary target mRNA, it is designed to inhibit, promote, or modify production of a specific protein to achieve its intended therapeutic effect. Ionis has been a leading innovator in the field of ASO therapeutics for the last several decades. So far, we have developed 3 commercial products, Spinraza, Tegsedi, and Reliver. We have 6 ongoing phase 3 studies with more than 30 other programs in early clinical phase 1 and phase 2. And we're expecting to have more than 12 medicines on the market by 2026. The SoloVPE method has already been implemented in a lot of these programs already. After the brief introduction, I'd like to switch gears to talk about why we need in-process assay control for ASO drug product manufacturing. The flowchart on the left shows a generic manufacturing process for an ASO product. As you can see, a number of in-process tests are performed throughout the manufacturing. In particular, the drug product assay is generally considered as a critical quality attribute. Therefore, it needs to be properly controlled in manufacturing. Despite ASO's relatively large molecular weight, it is still regulated as a small molecule globally. So regulatory agency would normally require the drug product assay to be controlled within 95 to 105% label claim from late-stage development to commercial. Therefore, we have to control the in-process assay in a tighter range to achieve this target in final drug product. Besides, ASO drug substance is known for being very hygroscopic. So rapid water absorption and desorption can occur during residual water testing, API packaging, and dispensing, which may lead to a significant discrepancy between measured moisture level and its actual level in the API. Having a proper in-process assay control thus can eliminate variation when calculated based on the API weight due to the uncertainty in its actual moisture content. Moreover, in-process assay check can also ensure the compounding process is correctly executed to allow us to detect and mitigate any major process deviation in a timely manner before moving forward to the final fill finish steps. We used to have an HPLC method in place as the in-process control. Recently, we have developed a new SoloVPE method and started to implement this new method as our ASO due to several advantages that I will mention in a later slide. In this slide, I'd like to briefly go over the principle of the SoloVPE system for those who may not be familiar with the technology. The SoloVPE system utilizes a so-called variable path length technology to measure a sample's UV absorbance by varying path length with a step motor controlled optical fiber rate. The path length can change from 5 micrometers up to 15 millimeters, which is a much larger range than the traditional UV instrument. Therefore, it can allow a direct measurement of samples absorbance for a lot of high concentration protein products. Sample concentration can be derived from slope of the linear curve of the sample absorbance versus path length. So based on the well-known Beer equation, the slope of the curve can be expressed as a product of extinction coefficient and sample concentration. If the extinction coefficient is none, then the sample concentration can be directly calculated. Alternatively, the sample concentration can be obtained by comparing its slope to that of a standard solution with a predetermined concentration. So we have taken this second approach in our method, as marked by the red star. This is because the standard is readily available, so we don't have to measure and certify the extinction coefficient for each ASO. Besides, measuring the standard and the sample at the same time can also help offset some of instrument variation. To develop a suitable in-process assay method, we did face a number of challenges. From drug product manufacturing side, obviously it would require the method to be able to turn around as quickly as possible, but it also needs to have excellent accuracy. Plus, it would be ideal to develop a one universal method for all ASOs. On the other hand, ASO has some unique inherent properties that can create hurdles for developing a straightforward SoloVPE method. Firstly, ASO normally has a very high UV extinction coefficient value in the range of 20 to 25 ml per mixed centimeter, which is much higher than most of the protein products. Secondly, ASO can also have enhanced intermolecular interaction in aqueous solution with increase of a sample concentration. So at a higher concentration, it can start to show nonlinear change of some physical chemical property like UV absorption and viscosity. In the very beginning of method development, we tried a very generic method applying all the default settings for the instrument, but found that such method didn't work very well for our ASO products. So we decided to take a more systematic quality by design approach. With the help of SME from Repligen, we did some brainstorming and came up with a fishbone diagram as shown in this slide to list all the relevant factors that could potentially impact the method accuracy and precision. These factors cover both sample parameters like sample concentration, sample preparation method, and dilution solvent, as well as instrument parameters such as sample vessel, wavelength, absorbance threshold, and so on. This fishbone diagram then became the roadmap to guide us for progressive method optimization. The first major factor we studied is the sample concentration. We investigated the impact of sample concentration on accuracy as shown in the left plot. The method accuracy was very excellent, below 1 mg per ml, but started to deviate from 100% above 1 mg per ml, and got even worse above 20 mg per ml. This is not that surprising. As I mentioned before, ASO has a very high extinction coefficient, so the sample absorbance above 20 mg per ml has already exceeded the upper limit covered by the linear Beer equation, even with the smallest 5 micrometer path length the SoloVPE is able to achieve. So while we think the deviation between the 1 and 10 mg per ml is likely due to the enhanced interaction between ASO molecule in the solution, given that the intended ASO drug product per concentration is normally above 20 mg per ml, so our study suggested the sample without any dilution cannot be measured very accurately. Eventually, we have chosen to dilute the sample to a universal 0.1 mg per ml to achieve a more desirable accuracy. So while this obviously adds one more step to the procedure, this dilution step can ensure any intermolecular interaction or any impact from the excipient is negligible, so that it can also work for our advantages to be potentially used as a universal method. This slide illustrated two examples of how we optimize other sample and method parameters. What we did is to monitor the slope variation continuously by taking 3 repeated measurements each day for a period of 3 days. Whenever possible, we try to study variation of multiple parameters simultaneously to capture any potential interaction between the factors. As shown in the left variability chart, absorbance threshold and UV averaging time was set at 1.0 and 0.25 seconds by default. Our study showed that the absorbance threshold at 1.8 and the UV averaging time at 1 second were chosen for the method, as they can provide a more stable slope reading over time, as indicated by the red star, compared to their default settings. In the right variability chart, large silicon vessel was chosen for providing a better precision than a small plastic vessel. Another study we did is to look for a proper standard for system suitability test. As we learned, a lot of CMOs, they have historically used BSA standard solution for their UV instruments. However, in our study, we found that the confirmed slope standard from Repligen provided a better accuracy and precision over a wider range of absorbance threshold, and therefore it is a better choice for the system suitability test. So eventually, we came up with an optimized method we consider suitable for our ASO drug product. As you can see from the table, each parameter in the previous fishbone diagram was optimized for achieving either better accuracy, precision, or simplicity. It is also worth mentioning that of these instrument parameters listed here, they are easily accessible from the control panel of the software. As I mentioned previously, we have been using a HPLC method for in-process assay testing. Compared to this HPLC method, the new SoloVPE method offers several obvious advantages. The HPLC method is known for being very accurate and precise, but it is also quite complicated to operate. It has 9 steps, and it can take up to 6 hours to report the result. In contrast, the new SoloVPE method only has 5 steps to cut down the turnaround time to 1 to 2 hours, which is more desirable for in-process testing, not mentioning the instrument itself is also much easier to operate. Moreover, we have also confirmed accuracy and precision are not sacrificed with the new method, as you will see from the following method validation results. To implement the new method for GMP production, we transfer and validate the method in several CMOs. This slide here shows the master validation plan according to the ICH method validation guidance to test the various method characteristics, including system suitability, specificity, linearity, precision, robustness, and sample stability. In particular, it's worth mentioning that the accuracy criterion is set to be within 97 to 103% to provide a 2% safety margin to ensure the final 95 to 105% specification for the drug product can be met. We've also verified a short-term storage stability of diluted sample at 5C and 20C. I will showcase some of the representative validation results in the following slides. In these slides, excellent linearity between sample slope and concentration was confirmed at a range of 50% to 150% of the nominal concentration for two different ASOs. This slide shows very good accuracy expressed in sample recovery for a variety of ASO drug products with different chemical modification, concentration, as well as in different excipients, confirming that the method is universal for ASO drug products. In this slide, intermediate precision by very different analysts and instruments was evaluated internally and as well as at another CMO. The percentage ISD was found to be well within the acceptance criteria of no more than 3%. To test method robustness, we made a number of intentional deviations to the normal procedure as shown in this table to see if any change would impact sample slope. We found that all these adjustments led to no more than 1% change comparing to the control, suggesting that the method is pretty robust against most common deviations, except for the situation when the sample cover was left open. In this case, the slope dropped significantly, along with a dramatic increase in the percentage ISD. I have to mention that keeping the sample cover open is a very obvious deviation and usually is avoidable and therefore shouldn't happen during a routine testing. This table compares the sample recovery results between Ionis and two different CMOs. I want to mention that when we initially transfer the method to our CMO site, we normally ask them to do a quick feasibility testing to mitigate any gaps in knowledge and practice before asking them to execute a full-scale method qualification. Once the CMO went through the initial learning curve, they were able to perform the method very smoothly and generate very reproducible results compared to our in-house data, as you can see from this table. Most CMOs have already accepted as a platform method. So after initial method of validation, we're able to apply the method directly for all our ASO in clinical batch production. After getting reliable results from classic ICH method validation, we continue to evaluate the method performance by exploring other tools. From a statistical point of view, any reported result from a method is equal to the sum of the true value, systematic bias beta, and random normal variation epsilon. The second beta term determines the method accuracy, while the third epsilon term determines the method precision. We utilize measurement system analysis, MSA, also known as gauge R&R, to further characterize the method variation. The bottom diagrams show the MSA study design. Basically, it's a DOE design to test each combination of common method variables, including 2 different test days, 3 different analyses, 3 different drug product lots, 2 different separate sample dilutions, and 2 repeated measurements. The order of these combinations was further randomized whenever feasible to eliminate the effect of any other uncontrolled variables. The top left chart shows the study result. It is a variation chart of sample recovery for each variable combination. A data distribution was derived from the chart and is shown in the top right plot. It was found that the results followed a normal distribution nicely with a mean recovery value of 100.3% and a standard deviation of 0.25%, suggesting the method provided a very good accuracy with a very small overall method variation, regardless of changing all these variables. In addition to the traditional mean value and the percentage ISD, we also use the 99%, 95% tolerance interval as another predictor for method variation, and it was calculated to be 99.6% to 11.1%, which is also well within the 97 to 103 limits we have set. We further analyzed the contribution of each variable to the overall method variation, as shown in the bottom left table. It was found that the test state appeared to be the biggest contributor, which indicates that all the other variables are in very good control, leaving the small total method variation mainly come from the inherent day-to-day drifting of the instrument. So through the MSA study, we were further assured that the method is well capable of fulfilling its intended use. In the future, we plan to further implement this method at all our CMOs to support drug product manufacturing of all stages. Meanwhile, we are developing a new method for residual ASO detection to support equipment cleaning validation. Besides, we are also exploring the feasibility of using flow VPE in our drug substance manufacturing as a PAT tool for inline concentration measurement. I'd like to recap my presentation in the following 3 bullet points. First and foremost, before initiating any method development work, I think it's very important to do a due diligence to get a good knowledge and understanding on the product, manufacturing process, as well as the instrument, so that we can set proper expectation on the method, and we can also decide on the level of accuracy and the precision we want to achieve for the method. Secondly, whenever time and resource are allowed, I would recommend utilizing a variety of quality by design tools in the method development and validation. Our experience has confirmed it's worth the extra effort, and we were able to get additional information and assurance on the method capability. My last and third point is that to take advantage of all the available internal and external resources, including technical support from Repligen SME and a variety of technical notes and the papers they have published. If the method will be transferred to a CMO or CRO, we can also leverage their knowledge and expertise. Utilizing these resources wisely will help us to develop and validate a suitable method in a quicker and more efficient manner. So last but not least, I want to thank my colleagues at Ionis as well as SME from Repligen for their great contribution to this work. So with that, I'd like to thank you all for your attention. I will stop here and happy to take any question you may have.
Thanks, Haiqing, for an excellent presentation. I'd like to remind everyone that you can submit any questions you have for Haiqing in the Q&A box on your screen, and we'll get to as many as possible. With that being said, let's dive right into the Q&A session. And our first question is the SoloVPE method able to match the accuracy and precision of the HPLC method you have used?
Yes, that's a good question. As I mentioned in the presentation, we have confirmed the SoloVPE method is able to provide a comparable accuracy as the HPLC method, with the error typically less than 2%. As for the precision, it's normally less than 1%, which may not be as good as HPLC, but still sufficient for the intended use. Overall, we think the SoloVPE is a better method than HPLC due to its simplicity and the quicker turnaround.
Mm-hmm. Excellent. Thank you. Okay, let's take a look at our next question. Understanding that Ionis has decided to adopt a method that includes dilution, what is the dilution process and how does it differ from the traditional method you were doing?
The simple dilution is done gravimetrically using an aluminum waveguide to reduce the variation during this step. The simple concentration calculated from the method is expressed in the unit of mg per gram and can be converted to the unit of mg per ml using the sample density.
Mm-hmm. Excellent. Thanks, Haiqing. Let's move on to our next question. Do you think the method can be used for other oligonucleotide molecules like siRNA or even much larger molecules like mRNA?
Yeah, it's a great question. So we don't have a direct experience working on siRNA or mRNA. That said, given the structure similarity between ASO and siRNA, I think the method can be directly used on a siRNA drug product without any issue. As for the mRNA, even though it has a much larger molecular weight, I think in principle it should be still doable to use the SoloVPE system for mRNA assay measurement, but some tweaks on test parameter may be required.
Okay, excellent. Thanks again. Let's move on to our next question. Do you think the technology would be applicable to drug substance manufacturing? For example, rapid concentration, determination of crude oligo solutions, and purification fractions without dilution?
Yeah, well, yeah, in this case, we're not using the SoloVPE for the inline measurement for your question, but in fact, that would be the FlowVPE system that we're also trying. It uses the same technology, but it is developed to be integrated into the process for real-time concentration measurement. I also want to mention that the FlowVPE is able to measure a much higher ASO concentration directly without a need to do a dilution. So it would be enabling a continuous process monitoring for drug substance reaction and purification steps.
Excellent. Thanks, Haiqing. Let's move on to our next question. I'm doing my research on an osteoporosis drug development using, it says herb. In which way can I utilize your suggestion, and how can I recommend this to my institution?
Right. I think I don't have a direct experience working on a herb drugs product, but I think if you can take the recommendation I gave in the three key takeaway bullet point. So first of all, you have to really understand the need for your product, and if you want to use the SoloVPE for your assay, you want to see how accurate, how precise you want it for the assay measurement, and and if you consider it's doable for your product, then you have to utilize all kinds of quality by design tool if possible, and to optimize the method. So I think we have to remember there's no perfect method we want to develop. It is a good method, good for its intended use.
Mm-hmm. Excellent. Let's move on to our next question. Can the SoloVPE method be used in materials other than drug products?
I would think so, because what we are doing at Ionis is mainly working on the ASO drug product. For that question, I don't know if I can defer to Joe, from Repligen to answer this question.
Mm-hmm. Okay.
Yes, the SoloVPE can be used in a variety of different other applications. We're not just limited to final drug product. It would depend where in the process you're looking to make a OD-based measurement, as long as there's an active chromophore and the previous method did not require any separation.
Mm-hmm. Excellent. Okay, let's move on to our next question. Will you elaborate on the time you spent performing SoloVPE method validation and transfer versus traditional HPLC validation and transfer? Where did you see the biggest savings?
Yeah, it took us much less time by saving at least 50% to transfer the SoloVPE method to a CMO site compared to the HPLC method. The work can be done in about 2 months if you have a well-written SOP and a method transfer protocol in place.
Excellent. Okay, let's take a look at our next question. And before I do that, I just want to remind everyone that if you have any questions for Haiqing, please type them in the Ask a Question box, and we'll get to as many as we can. So our next question is, I assume in this case, you already know the purity of your ASO. Otherwise, you are overestimating the assay by using SoloVPE. Is that right?
Yeah, that's a good question actually. Yeah, so we're using the purity of our drug substance for calculating the assay in the drug product. The reason is being that our ASO is highly modified, chemically modified, and with a high chemical and physical stability on storage and even in the solution. So we have a platform data showing that the manufacturing process would not impact the purity of the drug substance. So that allows us to calculate the drug product assay accurately.
Okay, terrific. Thanks again for your answer. Let's move on to our next question. Can you talk about the future applications you mentioned? In general, I'm interested in hearing more about how you plan to use the SoloVPE in line.
Right. I think I touched base on that question a while ago. So in this case, we're not using the SoloVPE for the inline. So we're going to be using the FlowVPE to utilize as a process monitoring tool for the real-time concentration measurement for our drug substance manufacturing.
Excellent. Thanks again. I'd just like to remind everyone that if you have any questions for Haiqing, to please type them in now. Let's take a look at our next question. What assay method do you use for final drug product release? Can you use the SoloVPE method for assay release of finished drug product as well?
Yeah, I think that's also another great question. So we internally we use our LCMS method for drug product assay release. But in my opinion, the SoloVPE method can also be used for final release, since it can be fully validated against ICH requirements for assay method validation. However, one caveat is that it's not considered as a stability indicating method. So you cannot use it alone in your specification, but also need to include another orthogonal stability indicating method like LCMS to monitor the assay stability on storage.
Excellent. Thanks again. Well, it appears that we've gone through all of our questions. I'd like to thank Haiqing for sharing his knowledge with us and also offer a special thank you to Repligen for sponsoring today's event. Please keep a lookout for an email containing a link to view this webinar on demand and to share with your colleagues. Thanks for attending the webinar, and please enjoy the rest of your day.
Thanks, Mike. Thanks, everyone.
Thank you.